on molecular pathology of differentiated thyroid cancer. I believe the purpose of this talk is to give you a general introduction to the field. These are my disclosures. So, and to talk about most common type of thyroid cancer, which is a well-differentiated cancer. Just to remind everyone that most of thyroid cancers develop from follicular thyroid cells, about 97% of them, and minority medullary carcinoma develop from C cell. And they're generally subdivided, if you're talking about cancers, into well-differentiated, which is a papillary carcinoma, follicular carcinoma. More recently, Hurthle cell carcinoma was added as a separate, not as a variant of follicular carcinoma, but as a separate entity because it has a distinct genetics, and we know that represent a separate type of thyroid cancer. So these are well-differentiated cancers that we are going to discuss right now. And then during the symposium, we'll definitely cover two less common but more lethal type of thyroid cancer, which is a poorly differentiated carcinoma and anaplastic carcinoma. So over the last 10 years, we also advanced our understanding of the precursor lesions for well-differentiated thyroid cancer. One of such lesions is NIFTP, or non-invasive follicular thyroid neoplasm with papillary-like nuclear features. We also briefly talk about it. It may have a less sort of impact on the oncologic aspect of thyroid cancer, but it's important for our understanding how thyroid cancer develops. Also want to mention that we live in the era of genomic medicine and genomic oncology. I mean, the knowledge in this area have been accumulating for a long time, but the explosion in the knowledge started about 15 years ago with introduction of next generation sequencing, which is a revolutionary technology that allows us to interrogate hundreds and thousands of areas of human genome in one reaction in a short period of time. This really technology led us to dramatically expand our knowledge on genetics of all human diseases, and of course oncology, and of course thyroid cancer. And that's again what we are going to discuss during the whole day of our symposium. So if to look overall at the molecular pathogenesis of thyroid cancer, it is relatively relatively simple. We know that the vast majority, almost all, at least papillary and follicular carcinomas develop through the mechanism by activation of two signaling pathways, MAPK, or mitogen activating protein kinase pathway, and PI3 kinase AKT pathway. And it's not necessarily a very new sort of concept. Researchers have been studying it for decades, but now we really have extremely deep knowledge, deep layer of knowledge, understanding how it happened and what secondary and tertiary event happened during this process. So, and what is important also to realize that activation of these pathways happens through a very different and multiple mechanisms and genomic alterations. It starts from the activation of one of the receptor tyrosine kinases, such as RET and TRK. Then there are cytoplasmic kinases, such as a BRAF, or sort of other mediators, such as RAS, and so on and so forth. So it is important, and we'll be repeating it multiple times during this symposium, to keep in mind that there are three main mechanisms how driver alterations occur in thyroid cancer. It's point mutations, such as RAS, BRAF, gene fusions, and copy number alterations. Each of these events may serve as a driver. It means that something that can initiate and force the cell to divide and become cancerous cell. we discussed it, already a little bit of yesterday, but I want to repeat because I believe we have a new audience today, and this is also very important. This is the TCGA studies that showed the molecular profiles of about 400 papillary thyroid carcinomas. So it highlights that about 75% of the tumors develop through the mechanism of point mutations, such as BRAF or RAS. About 15% have gene fusions, and a number of them have copy number alterations, and in some situations it's a solitary or unique event. It means non-overlapping with point mutations or gene fusions, and likely represent a driver event in thyroid cancer. However, I want to spend a little bit of time discussing this sort of additional kind of additional data set of about 500 cases of papillary thyroid carcinoma. I think that the importance of it is that, first of all, we now 7-8 years after TCGA, we know some new additional alteration, and secondly, it may represent more accurately the proportion of different mutations in thyroid cancer, which was somewhat biased in the TCGA cohort because of nature how it was built. So let's look one more time. About 70% of papillary carcinomas develop through the mechanism of point mutations. So most common event is BRAF, 34%. Overall, I mean, it would vary from 30 to 70%, depend on the institution and country in the world. But overall, it still remains the most common molecular event in papillary thyroid carcinoma, BRAF V600E. This is an early genetic hit. So we are going to talk about early and late, because that's typically how sort of particularly aggressive thyroid cancer develops. So we already discussed that 3 genes, NRAS, HRAS, and KRAS, represents the second most common event, which can be found in about 20% of papillary thyroid carcinomas. More recently, after TCGA, we learned about few other important molecular events. For example, one of them is Dicer1, a gene that was relatively recently discovered that participates in processing of microRNA. So now we know that about 2% of all PTCs have these Dicer1 mutations, which can be germline and can be actually sporadic and be germline. So it's important. This is an additional sort of situation when thyroid cancer may develop as part of the inherited disease, and we briefly touch on it later in my talk. Also, just briefly mention that some other very rare mutations that are more recently have been identified are, for example, MEK1 or MEK2K1 mutations, SPAP mutations. So again, we are getting more and more understanding, but now we are at the stage when we know molecular composition of about 97% of papillary thyroid carcinomas. So we still keep discovering new targets, but they constitute very small sort of proportion of remaining unknown, genetically unknown papillary thyroid carcinomas. So these are all early genetic hits or early events. Late hits are very important because they are associated with more aggressive behavior of PTC, and those include TERT, found in about 10 to 12% of PTCs, P53, PIK3CA, and one more marker is AKT1. These are secondary hits that typically seen in poorly differentiated anaplastic carcinomas, but when they are seen in papillary carcinomas, typically we perceive them as markers of more aggressive behavior of these tumors. Moving to gene fusions, about 15 to 20% of thyroid cancers develop through the mechanism of gene fusions. They generally can be subdivided into two classes, into receptor tyrosine kinases or RTKs. Very high importance of understanding and recognizing such class of gene fusions is because they represent an excellent therapeutic target. which will be extensively discussed during the course. So the most common of RTK fusions is red gene, also very important, NTRK and others. So in terms of the non-RTK fusions, SADA fusion is also more common, as well as PAX8 PPR gamma fusion. And finally, copy number alterations. About 20% of PTCs have this molecular event. It can be of two types, so-called, Hurthle cell type or non-Hurthle cell type, and we also briefly discuss it later in my talk. But this is again that the approximate prevalence and composition and molecular composition of papillary thyroid carcinomas. TCGA paper taught us another very important piece of information about PTCs. Now we know that all PTCs can be generally divided into two molecular groups, into BRAF-like or BRAF V600E-like and RAS-like. It is very important to recognize these two classes or two subgroups of papillary thyroid carcinomas, because they have very different biological and clinical behavior. So these tumors morphologically are separated, can be predicted and can be sort of looked and are associated with different phenotypes. RAS-like tumors are mostly follicular pattern tumors, so follicular variant of papillary carcinomas, whereas BRAF-like tumors are classic PTCs or tall cell variant. But most important thing is that they have very different levels of expression of thyroid differentiation markers, which we discussed extensively yesterday, which is a pretty high level of expression in RAS-like and loose of expression of NIS, TSHR, and other critical genes responsible for thyroid differentiation and iodine absorption in BRAF V600E tumors. Also briefly mention that most other alterations can also fall into either RAS-like or BRAF-like subgroup of papillary thyroid carcinomas. What about follicular thyroid carcinomas, the second most common class of thyroid cancer? They are RAS-like, obviously, because they are follicular. And if one look at follicular thyroid carcinomas molecular profile, it lacks BRAF mutation and is dominated by RAS and RAS-like molecular alterations, either mutations or gene fusions. The molecular profile of FTC is similar to follicular adenomas, again highlighting that many FTCs develop from follicular adenomas, and it's also similar to encapsulated follicular variant of PTC, and the type of papillary carcinomas that is composed of follicles, but has nuclear features of PTC that is not seen in follicular tumors. And then classic papillary carcinomas and infiltrative papillary thyroid carcinomas have very different molecular profiles. So really, if we take papillary carcinomas and follicular carcinomas and put them together and split them biologically, the split will not be between PTC and FTC. It will be between follicular type, follicular pattern tumors that are RAS-like and more papillary, classic papillary type tumors, which are BRAF V600E-like tumors. So a few words about the NIFTP, or non-invasive follicular thyroid neoplasm with PTC-like nuclear features. This is believed to be a precursor for encapsulated follicular an invasive follicular variant of PTC. These tumors were called as PTC before reclassifying, and now it's believed to be a precursor. If overall we look at development of papillary and follicular thyroid carcinomas, So for classic PTC, it is general beliefs that most of them develop from papillary microcarcinoma. So these are BRAF-driven tumors that from very early stage become cancerous, but they have different potential to grow and really develop as a clinically relevant, impactful tumor. Then for follicular growth pattern tumors, there is a sequence of events from developing from follicular adenoma to follicular thyroid carcinoma. And when nuclear features of PTC present, from NIFTP to invasive encapsulated follicular variant of papillary carcinoma. So one more time to summarize, because this is really very important. What are the BRAF-like and what are RAS-like tumors? And why it is important clinically to recognize this molecular subclassification of these tumors. BRAF-like tumors are classic papillary carcinomas and infiltrative follicular variant of PTC. You see it called follicular variant because it composed mostly follicles, but there is a highly irregular border because this is infiltrative tumors, and these tumors are also typically have BRAF V600E mutations. Like I said, these tumors have classic nuclear features of PTC. They are all infiltrative. Very importantly, clinically, they spread to lymph node first, to regional lymph nodes, and then to later to distant sites, and they lose expression of genes associated with thyroid differentiation and iodine absorption from early stages. RAS-like tumors are follicular adenomas to follicular carcinomas when invasive, NIFTP, and invasive encapsulated follicular variant PTC. This tumor was probably developed initially as NIFTP encapsulated, that you see is broke through the capsule and invaded most of the lobe of the thyroid. But look how smooth the border of this tumor is. So these tumors do not provide infiltrative growth. They grow pushing into the adjacent normal parenchyma, and importantly, they invade blood vessels, and if they metastasize to distant sites, and extremely rarely in lymph nodes. Again, they encapsulated tumors, and important that they retain expression of genes associated with thyroid differentiation for a long time. Few words about kinase fusion positive thyroid carcinoma. Again, this becomes a very hot topic in the field because of availability of inhibitors, specific inhibitors. So how we can morphologically predict that certain tumor may carry a fusion such as NTRK or RET or ALK fusion. Very nice recent paper published from MGH showing that, suggesting that tumors that are positive for this receptor tyrosine kinase fusions, they are characterized by multinodular growth, prominent fibrosis, and extensive lymphovascular spread. They also suggest that when you see these morphological features, you have to have a higher index of suspicion for the presence of the gene fusion in this tumor. In our experience, we do see all these three features, and I would like to add one more, which is the mixed papillary follicular and solid architecture. This is a nice example from the paper. You see this tumor has, in addition to extensive fibrosis and multinodularity that was mentioned before, also, look, in one area it has very papillary growth pattern, and another it has a more solid growth pattern. In the third area, more follicular growth pattern. When we see this combination of these different growth patterns in the same tumor, in my experience, we really have to start thinking about gene fusions, and we will find them in a significant proportion of cases. Now, a few words about Hurthle cell carcinoma. As we discussed before, this is now a separate type of thyroid neoplasm. I mean, you know that morphologically, this tumor is characterized by being composed of cells with a lot of cytoplasm, which is very granular due to accumulation of numerous abnormal mitochondria. We know that these tumors have mitochondrial DNA mutations, which may not be carcinogenic. Some of them have nuclear DNA mutations, but the hallmark of these tumors are copy number alterations of very peculiar and specific type, called neohaploid type of chromosomal copy number alterations, which is either loss of the entire chromosome, you see, instead of 2 chromosomes, many chromosomes present in single numbers, or presence of 2 chromosomes, but both of them belong to the same allele, either maternal or paternal. So 1 chromosome is lost and second is reduplicated. And now we have the two chromosomes of the same type, either paternal or maternal. This is really a very enigmatic type of genetic alteration. We're still learning to understand how it caused cancer development, but there is a strong feeling in the field that this represents a driver, an early driver event in Hurthle cell carcinomas. So is this event, copy number alterations, specific for Hurthle cell carcinomas? The answer is no. Although about 80% of Hurthle cell carcinomas have this copy number alterations, also a subset of Hurthle cell adenomas also have such alterations, but not Hurthle cell hyperplasia seen in Hashimoto's thyroiditis. Is it surprising? No, because again, Hurthle cell carcinomas unlikely develop from 1 or 2 millimeter tumors in cancer. Many of them grow as adenomas, and at some point in time, acquire probably additional genetic or epigenetic alterations, get malignant invasive potential, break through the capsule, and develop as a garden variety Hurthle cell carcinoma. Why it is important? Because we have to realize that some of these histologically benign adenomas are precursor lesions for cancer, and patients may benefit from removing such tumors at pre-invasive stage. So just one slide on medullary carcinoma, just for the sake of to cover all well-differentiated cancers. These tumors, as you know, can be familial or sporadic. In sporadic tumors, about 60% of them have red mutations, sometimes indels, but mostly point mutations. About 20% have RAS mutations. And there are some very rare occasional sort of BRAF non-V600E mutations and some few other alterations found in these tumors. So to summarize, when we look today in 2022 at thyroid cancer, at differentiated thyroid cancer, and when we look at 3 types of events, at point mutations, at gene fusions, and at copy number alterations, we pretty much can cover most of the tumors in this category, at least of well-differentiated thyroid cancer. So we have a tremendous amount of knowledge about the genetic landscape of thyroid cancer, and now it's time to move this knowledge to the patient bed and start managing these patients more individualized, based on the information that we can get from genetic profiling. Couple words about few smaller but important more recent developments in the field. I mentioned already that about 1 to 2% of all thyroid cancers have DICER mutation. DICER mutation may be an indirect evidence of DICER1 syndrome that is very peculiar and has a number of very rare, unusual tumors, such as pulmonary pleuroblastoma and several others, Sertoli-Leydig tumors, and so on and so forth. And typically in younger patients, and when patients grow over the age of 20, practically the only organ that can be affected frequently is the thyroid. What is really interesting about these tumors, there is a spectrum of thyroid cancers driven by DICER1 mutation. On one end of the spectrum is follicular adenomas and PTCs or FTCs. And these tumors characteristically have extremely abundant colloid. These are all pictures of representative papillary carcinoma or follicular carcinoma, tumors driven by Dicer mutation. And if you look at it, there is so much colloid presence that it's almost difficult to believe that this is this cancer, but there are such colloid-rich macrofollicular variants of PTC or FTC, and again, they characteristically have Dicer1 mutations. On the opposite spectrum of Dicer1 driven thyroid cancers are poorly differentiated cancers of childhood and adolescence. They are very rare, but we and others have recently shown that approximately 80% of all poorly differentiated carcinomas developing in children and young adults up to 20, 30 years old, they are driven by Dicer1 mutation. Also a rare disease, but it's a very different from all other sort of classical poorly differentiated carcinomas that we see in adult patients. And finally, a few words about tumors, which is called hyalinizing trabecular tumor. This tumor has nuclear features of PTC, and therefore it's on cytology, it's frequently diagnosed as suspicious for PTC or PTC. And it has been like 20 years debate if this is a variant of papillary thyroid carcinoma or not. Now we know beyond any doubts. Why we know that it's not a variant of PTC? Because we and others have shown more recently that these tumors have a very specific and different type of genetic alterations, which is a fusion between PAX8 gene, very well-known thyroid transcription factor, and GLIS fusions and GLIS gene. So there are two types of fusions, PAX8 GLIS3 and PAX8 GLIS1. As you can see, most of the hyalinizing trabecular tumors have mostly PAX8 GLIS3 fusion, and this fusion are not present in papillary thyroid carcinoma. So really providing strong evidence that these are two separate types of thyroid cancer, and laying into rest all this debate about if this is a variant of PTC or not. So to summarize what we discussed today, if we want to talk about molecular classification of follicular cell-derived thyroid cancer, first of all, I want to say that the fundamental biological difference between hyperplastic nodules, most common type of nodules within thyroid, and tumors is that hyperplastic nodules is a non-clonal event. It doesn't develop from a single cell, and therefore it lacks any genetic alterations. And all types of thyroid cancer, they have one or another driver mutations. Also want to remind us there are three main classes, three main groups, three main molecular groups of thyroid cancer. We can talk now about thyroid cancer as a BRAF-like, RAS-like, and copy number alteration driven thyroid cancer. BRAF-like tumors are typically classic papillary carcinomas that develop from microcarcinoma. RAS-like tumors are follicular carcinomas, many of which develop from follicular adenomas, and invasive encapsulated follicular variants of PTC developing from NIFTP. This is a RAS-like tumor cloud. And copy number alterations driven typically Hurthle cell carcinomas that develop from Hurthle cell adenomas. So this molecular classification is important because, again, these tumor types have specific clinical presentation and morphology. So to conclude, as of today, we know genetic driver events in the vast majority of thyroid cancers. Such driver molecular events have strong correlation with phenotypical features, biological and clinical properties of differentiated thyroid cancer. And therefore, as of today, we have to start talking about molecular classification of thyroid cancers with implication for the management of these patients. Thank you for your attention.